This is a
thank you, Galen. Do people hear me? Okay, good. Louder? Closer to the mic? Is that working now?
Yes.
Okay.
Maybe you could lift the mic a little and then
Is that better? Yeah. So we come here a couple of weeks every summer, and so it's really exciting to be able to speak to the people who live here or spend time here, and to be invited here. And thank you, Galen, for inviting me. And also my kids Seth and Leia wanted me to mention them, so I wanted to mention them. They're here. And they promised to be very quiet during the talk. Right, so I'm gonna, I decided not to give exactly my normal talk that I've been giving in the last few months, and so it's, and to really give you a flavor of what's this field is like, and what's changing in this world, and what's in this book, which attempts to be a summary and a channel through which you can learn about what's happening. So what I'm going to talk about is 3 lessons in how we as a community were wrong, and I'm just going to go through the 3 lessons, and then maybe there'll be some time for talking as much as people want to talk. There's time. So in 1960, about 60 years ago, Luca Cavalli Sforza, who is the person who really started this field of genetic studies of human history, made a grand bet. And he was a really visionary scientist who saw, really before almost anyone else, the power of learning about the past from the genetic data from people live today. And his idea was that we might be able to reconstruct the deep past, the shared past of peoples living today, based on patterns of genetic difference amongst peoples living today. And to make that idea work, because all he had data from was people living today, he had to make an assumption. And the assumption was that the people living in each place in the world today are pretty directly descended often from the people who lived there a long time ago. And so, for example, he analyzed the thin genetic data he had available to him at the time, beginning in the late 70s, and then this is an updated map from the 90s, where he looked at patterns of genetic variation across many places in our genome in Europe, and found that the biggest gradient of variation that was observed was in a southeast to northwest direction. And he interpreted this as the effects of the movement of farmers into Europe, which based on the archaeology we know occurred between 9,000 and 8,000 years ago, first from Anatolia, probably from present-day Turkey, landing in Greece and spreading from there. And his idea was that what we're seeing is the dilution of farmer ancestry as it moves southeast to northwest. But today, in light of ancient DNA, we know that the truth is like this in terms of the proportion of farmer ancestry, with the greatest proportions being actually in southwestern Europe, not southeastern Europe, and the dilution and the gradient being almost in a perpendicular direction. And this is not because the movement of farmers into Europe didn't happen, it's actually because so much happened since that time that overwrote those earlier events. And so really it's very difficult to reconstruct the past based on people today. The indigenous people of the world today, in fact, are people who replaced, in most cases, previous indigenous peoples in the same places. And I'm going to give you 3 examples of how orthodoxies or assumptions that were in place before genetic data have yielded to genetic data. And so what's really changed, and really changed beginning in 2010 and explosively changed really in the last few years, is the availability of this new technology of ancient DNA. The ability to extract whole genome sequences of the same quality essentially as we obtain when you sequence your genome for a medical genetics to screen for diseases, from ancient bones thousands, tens of thousands, in some cases hundreds of thousands of years ago. And so it's a miracle that this is possible. And I'm going to tell you a little bit about the technology that makes this possible. So you start with a bone, a skeletal remain, a tooth, some type of human remain. And in a clean lab, this is our clean room in Boston, where the goal is to protect the skeletal remains from the people who are handling the skeletal remains, who have so much more DNA than is in the bone, so they can easily contaminate the bone. We drill a small hole beneath the surface of the bone, try to extract powder that's less contaminated by the people who handled the bone. We take the powder, we release the DNA in a watery solution, and extracting and separating it from the protein and mineral content. And we sequence it in one of these modern sequencers that really became available in the late 2000s and dropped the price of sequencing literally by $1 million over a decade and a half. And that's made it possible to ask and answer questions about the past that were simply not possible to answer before. The first ancient genomes were published in 2010, and there were 5 of them. The number reached 10 in 2000 and above 10 in 2013, above 100 in 2015. And now there are about 1,700 ancient genomes. This slide is out of date. In our laboratory, there's close to 5,600, many of them unpublished. And I know that's true of other laboratories. And it's because of this amazing technology of ancient DNA. And the measure of how powerful a new technology is that when you use it to look at something that's not been looked at before, it reveals to you things that are new that nobody even imagined. It's a little bit analogous to what happened in the 17th century with microscopes, when for the first time people looked into the world of cells and saw things that they just didn't know before, that there were cells, that there were microbes. You can look at a flea and see it all big blown up, and there's marvelous pictures from the people who first did that, drawing the amazing things they saw that were very surprising to the worldview that they had. So I'm going to tell you the theme of this talk was 3 surprises. And the surprise one is that we're not all African. So there was a powerful consensus, which I actually was weakly a part of, that culminated in the late 80s and 90s, which is that all people living today are descended from people who spread out of Africa between 100,000 and 50,000 years ago, and all our ancestry comes from that. We know from the archaeological and anthropological record that there were other humans living outside of Africa, these big-brained Neanderthals and other humans who lived outside of Africa. But genetically, the data that we had available at the time showed that all lineages living today, persisting today, descend from are nested within variation that still exists in Africa. The variation of non-Africans is basically or nearly a subset of that in Africa. And based on this, and also the evidence of the oldest human sophisticated tool use being in Africa, there was a consensus that Africa was the homeland of the last spread of modern humans between 100,000 and 50,000 years ago. In 2006, it became clear to a scientist working in Germany, Svante Pääbo, that it would be possible to extract whole genome sequences from Neanderthals. These are these large-brained humans who were first discovered in the 1850s and 1860s in Europe, and who lived in Europe and Western Asia. They were as tall as us, they had brains as large as us, and they made very sophisticated stone tools like our ancestors did. And the question was, who were they? How did they relate to us? How closely related were they? And so I got invited in to collaborate with this group at the time when it was clear that they would begin to be able to obtain whole genome data. So Neanderthals were spread over the range shown in the top. That's Europe and Western Asia. And that modern humans were spread in Africa until about 50 to, or Africa and the Near East until about 50 to 50,000 years ago, after which point they exploded out of Africa and reached different places in Eurasia, mostly by 50,000 years ago, and then into the Americas at least by 15,000 years ago. So a question that has always been in people's minds was, as these modern humans exploded out of Africa, they encountered these archaic humans who had lived there for actually almost 2 million years. And a question was, did they interbreed? How did they interact? Today we're alone on this earth. We are not related within 5 or 6 million years to any other populations of animals living on earth. But at that time there were many groups of humans quite closely related to us relative to how we are related today. And these people must have met each other. In southern France, there are sites where there are layers of Neanderthals, modern humans, Neanderthals. They clearly interacted with each other. There have been novels written about this interaction, from the very popular novels like Jean Auel's novels like Clan of the Cave Bear. There's a novel by William Golding called The Inheritors, imagining this dramatic interaction between these people who can make incredibly sophisticated tools, but very, very different from each other. So the question was there interbreeding? Was there interbreeding in the Near East, where there's some evidence of Neanderthals, or in the homeland of Neanderthals, which is probably Europe? And what happened in the movement of humans into the less well-charted archaeologically part of Eurasia? So what I and my colleagues were involved with developing was a set of tests where we were all really trying to test whether Neanderthals interbred in some way with modern humans. And the way we did it was the following. There were multiple tests, and here is the simplest one, which is pretty easy to explain. So this is a cartoon of a chromosome, your DNA, which, as you know, is a 3 billion letter long, approximately, sequence of letters, A, C, T, or G, adenine, cytosine, thymine, or guanine, is packaged into 23 pairs of chromosomes, which are just packets of DNA that get passed down from father to child or mother to child. And this cartoon is a cartoon of a chromosome with this ball in the middle supposed to be the centromere, which is a structural element in the chromosomes, and these arms being the arms where the DNA is held. And so what we did is we lined up a chromosome from a European person and a sub-Saharan African person. This is a Nigerian individual. We looked at all the places we could detect where those two individuals were different. And then we looked which the Neanderthal had. Did the Neanderthal match the Nigerian person more or the European person more? And if it's true that we all come out of a common African population that separated earlier from the ancestors of Neanderthals, all people today should match Neanderthals exactly equally often, and you can show this mathematically, but it also makes sense intuitively. And when we actually did that analysis and said how often does the Neanderthal match the European or the Nigerian, it was about, we would expect it to be exactly equally often, but there was about a 2 to 4% significant excess matching to Europeans. So this was highly statistically significant. It wasn't just Europeans, it was also East Asians, and it was also New Guineans. It was all non-Africans, showed this pattern of excess matching compared to Nigerians, but not just Nigerians, to Central Africans, to East Africans, to Southern Africans. It was a general difference in non-Africans being more closely related to Neanderthals than are Sub-Saharan Africans. This was one of several tests, but using this combination of tests, we were able to show unambiguously that Neanderthals interbred with modern humans, and in fact, the Neanderthal sequence we had was from an individual related within, at most, about 100,000 years to the population that interbred with actually us, or at least non-Africans today. We also were able to estimate how much Neanderthal ancestry there is into people today, and the way we did that is as follows. We took a non-African and we saw how often they matched a Neanderthal relative to an African person, and then we replaced that with a second Neanderthal sequence we had data from, and we say how much of the way toward a complete Neanderthal is a non-African person, and they were about 2% of the way, and that we can interpret that as evidence that non-Africans today have about 2% of their DNA derived from these archaic humans. East Asians, we now know, have actually significantly more Neanderthal DNA than do West Eurasians like Europeans, and we know exactly why that is. I'm happy to talk about that as well. So what explains these patterns? One possibility is admixture somewhere in the Near East, in the common ancestral population of East Asians and New Guineans and Europeans, and that population spread throughout, and that's how this interbreeding occurred. And another idea is that actually there's some kind of ancient population substructure where sometime before the spread out of Africa, there were populations in Africa more closely related to Neanderthals and less closely related to Neanderthals. And that the population that migrated out of Africa was one more closely related to Neanderthals. And that's why non-Africans are more closely related to Neanderthals than are Africans. But that we can show that that's not true because we can show chunks of the DNA that are, as I said, are related within about 100,000 years. We also were able to estimate a date when this mixture occurred, because here again are cartoons of these chromosomes, stretches of DNA derived, which are the packets of DNA that we inherit from our mothers or fathers, 23 pairs of them. So here's supposed to be a pair of chromosomes from population A in red, which might be Neanderthals, population B, which might be modern humans. One generation later, you might have one of your packets on your chromosome 3. The one from your mother might be from a Neanderthal. The one from your father might be from a modern human. So what we do when we produce a sperm or an egg is we mix or match the chromosomes we got from our mother and father 1 or 2 breaks per generation. And what you do is you chop up your DNA until today, many generations later, we have a salad, a mixture, a mosaic of DNA from our mothers and fathers. And by looking at the dice size of that salad, the chunk size of the DNA from Neanderthals and modern humans, we know exactly how long it's been since the mixture occurred. And in this way, we can learn that the mixture occurred about 50,000 years ago. And in fact, we now have ancient DNA from a 45,000-year-old human from Siberia who has huge chunks of Neanderthal DNA that I'm circling here with my little arrow. Little long stretches that extend maybe a 20th of the length of a chromosome and must be within 20 generations or so of an interbreeding event with a Neanderthal and that individual's ancestor. Since that time, we've actually found an individual who's within 4 to 6 generations of interbreeding. So we can actually learn from ancient individuals who, with radiocarbon methods, we know exactly what their date is, how close it's been since the Neanderthal interbreeding was, and we now know that the main interbreeding event occurred in a fairly tight time window between 54 and 49,000 years ago, and is shared in all people living today. So the conclusions is that there's an interbreeding event in the common ancestral population that's spread around. While we were finishing the Neanderthal project, there was a finger bone in a cave in Siberia, southern Siberia on the border of Mongolia and Kazakhstan and Russia, that was marked as a modern human by the Russian archaeologists who excavated it. But in fact, when it was sequenced by this laboratory in Germany, it was found to be neither a Neanderthal nor a modern human, but something else entirely. It was a finger bone of a little girl. This is the size of it. And it was amazing that it was even identified as human. But once it was sequenced, it was clear it was like nothing that we had ever seen before. The individual here's several Neanderthals, was more distant from Neanderthals than any that were from each other, but genetically was more similar to Neanderthals than they are to modern humans. So this is a very interesting observation, and meant we had discovered a third population of humans, and a second population of archaic humans. We did the same thing, where we checked whether the Denisovan genome from Denisova cave matched some humans more than others, and this was a particularly interesting finding when we compared this ancient genome to a Chinese genome and to a New Guinean genome. We found that the Denisovan individual matched New Guineans significantly more often than it did the Chinese. And the only way that this could occur is if there had been Denisovan-related interbreeding into the ancestors of people in New Guinea today, and we estimate that it contributed about 4 to 6% of the ancestry of people in New Guinea, on top of the approximately 2% Neanderthal ancestry in this population. So this is an extraordinary finding. Not only is there another ancient bone from another population, but once we have it in hand, it reveals interbreeding events that we didn't know about. If the 2 times we've discovered archaic humans from DNA, we find 2 interbreeding events, who's to say that in the 3rd archaic human we find, we might not also discover further interbreeding events? This Denisovan genome was particularly amazing for us and really opened up the Pandora's box of what ancient DNA could show, because with Neanderthals, we knew that they were interesting because the skeletal record was incredible, right? It's been around for 150 years. These are people who made amazing stone tools that were incredibly difficult to make. For example of what these Neanderthals could do is, it used to be maybe 800,000 years ago, or 1.8 million years ago, that humans would take a kind of block of nicely chosen stone and whittle it away into the shape of what they wanted. But beginning about 300,000 or 400,000 years ago, in both the lineages leading to Neanderthals and modern humans, and maybe invented in the common lineage, there was an invention where people were able to choose a block of stone, and instead of whittling it into the shape they wanted, they turned it into what's called a core, a block that when hit just the right way with a soft deer antler or a harder object would pop out exactly the stone they wanted. And then when they hit it again, it would pop out another tool exactly as they wanted. A little bit like a sculptor who sees inside a block of stone exactly what they want to see. These people, both the Neanderthals and the modern humans, had physical intuition sufficient to envision the grain of these stones and see what would form. It's no surprise that these people were capable of all the things we're capable of doing today, but it was already in them at that time. These Denisovans are more closely related to Neanderthals than are modern humans, and surely they had many of the same innovations. But in their case, with the Neanderthals, we knew exactly what they looked like from their skeletons. Big-brained individuals made amazing tools. In Denisovans, we had no idea about them before the genetics. In Neanderthals, it was fossils which we knew it would be interesting to obtain a genome sequence from and ask questions like, was there interbreeding? With the Denisovans, the question was, it was a genome in search of a fossil. We don't know what they're connected to. All we have is this tiny finger bone. It's not a skull. It's not a full skeleton. We don't know what they looked like. We don't know how big their brains were. We don't know what kinds of foods they ate. We don't know what kinds of tools they make. But there's all these enigmatic fossils from East Asia, from China, from Southeast Asia, from the Philippines, from South Asia, from Siberia, from Korea, from Japan, that have not been sequenced yet, and it seems likely that some of them will be Denisovans, especially since some of them also interbred with the ancestors of New Guineans. I'm going to skip this slide, but the interbreeding of Denisovans not only occurred with New Guineans, but with the ancestors of Aboriginal people from the Philippines and from Australia. And if you look in these black shaded circles in these pie charts, this is where the Denisovan ancestry is present today, highest frequency in the Philippines and in Australia and New Guinea, and also in people who have mixed with New Guinean populations as they spread out into the Pacific and into Indonesia. There's also a little bit of such ancestry throughout East Asia, and we don't know whether that's coming from the New Guinea region or whether it's due to separate mixture events on the mainland. In fact, we know there is at least one separate mixture event in the mainland. So the conclusions here is that there was an initial interbreeding event of Neanderthals on the gateway of Africa, about 2% of the ancestry of non-Africans, and a further interbreeding event that led to Denisovan ancestry. And now we know of many interbreeding events amongst archaic humans and modern humans. I'm not going to go into all of them because each one of these is its own sort of struggle to figure out how it happened and explain, and I wanted to move on to the second part of my talk. In the second part of my talk, sort of another example where it was a great surprise. I came from Luca Cavalli's Sforza school where we thought we all came out of Africa, there was no interbreeding, and I was very much part of that school. My PhD supervisor was part of the group that established that, and when I came into the consortium, my very strong assumption was that that hadn't occurred. And my colleagues and I worked extremely hard, harder than we otherwise would have, to make the result go away because we were very skeptical of the idea that interbreeding had occurred. But the data seemed to show it in every possible way, and we kept developing new tests, and eventually it's just unambiguous. Right now, there's just so many signals and signs of Neanderthal DNA evident in our genomes. If you're a non-African person, it's just extremely obvious at this point. And so the data forced our hand in that case, even though our assumption was that something different was going on. Okay, the second thing I wanted to talk to you about was another case where I think a lot of assumptions we had coming into what I had when I grew up about the relationships amongst human populations and the nature of human population structure today are fundamentally wrong. So I think a lot of us have an idea that the population structure we intuitively understand in the world today, people grouped into groups like people like New Guineans or different groups of Africans or Caucasians or East Asians or South Asians, reflects some kind of age-old population separation that's happened for eons, right, for a very long time after a bifurcation and spreading from some common ancestral population. But that's profoundly wrong, in fact. In fact, 10,000 years ago, that structure would have been unrecognizable. And I'm going to tell you why that is, because we now know it's that so from ancient DNA. And the example I'm going to give is West Eurasia, which we know best because ancient DNA has been richest from West Eurasia, not because it's a particularly important place in the world, Europe, but because that's where the first ancient DNA studies have been, because scientists who invented this technology were largely in Europe, and that's where we have our best data. But everything that we look at suggests that the same sorts of patterns will be observed in other parts of the world as well. So I'm going to tell you a little bit about a story we published in 2016 where we put together new data from 44 individuals from the ancient Near East. So this is places like Iran and Levant, Jordan and Israel, Anatolia, and the northern part of the Levant, which is, for example, of the Near East, Caucasus, Armenia. So we combine that data with previous data from the steppe north of the Black and Caspian Sea in present-day Russia. and also many samples from Europe that we had data from to try to understand the nature of the human population structure that existed over the last 15,000 years, which was the period over which we were able to interrogate whole genome sequences from this time. And so this picture shows the different places, the Levant, Iran, Anatolia, present-day Turkey, the Caucasus, where Armenia is, and mainland Europe, and the steppe, where each of which we had samples at different points in time. And so the first observation we had, what we did is we took samples that we had at each time slice. So this is, on the top, is the earliest time slice, about 10,000 years ago. The Near East is a particularly important place in the world because it's where farming was first invented in the world. So farming was first invented about 12 to 11,000 years ago in the Near East, a little bit before it was invented independently, probably in China. And it spread from each of these invention centers in lots of different directions. So we looked at the data we had from Western Eurasia about 10,000 years ago at the dawn of farming, and we found that the average level of difference between populations as measured by the square of the frequencies differences between populations at that time was 10 times larger than what it is today. And the number on this scale is around 0.1, and that's the level of difference between Europeans and East Asians. So if you look in West Eurasia, there are 4 different populations at that time, and there was a collapse of that differentiation by about 6,000 years ago to a third of its level, and by 4,000 years ago to the present-day levels. So somehow something had happened to collapse the population structure in this region that existed 10,000 years ago, which harbored as diversity, as different as Europeans and East Asians, to the sameness that you see today. People call these people Caucasians or whites because people sort of approximately look the same and genetically are pretty similar compared to, for example, the difference between Europeans and East Asians. But in fact, this didn't exist 10,000 years ago. These people were not like that. And what you can see from the genetic data is the following. This is what happened. 10,000 years ago, we actually have real genetic data, whole ancient genomes of similar quality to modern genome sequence for medical studies from 4 populations. Iranian farmers who lived around this time, Levantine farmers who lived in Jordan and Israel, Western hunter-gatherers who lived in the far west of Europe, Eastern hunter-gatherers who lived in the far east of Europe. Each of these groups was as different from each other as Europeans and East Asians are today. And what happened is that none of them disappeared, but they all mixed each with other profoundly. And what you actually see in this region today is mixtures in different proportions of these 4 source populations with small additional contributions from other groups, but these are the primary sources of ancestry across this vast region. And it's this blurring that creates this relative sense of sameness that we see today. So this is a critique of people's picture of, quote, racial structure today that I think was not really in people's heads before. There are many good critiques of that, but one of them that was not in people's heads was the idea that 10,000 years ago, if you had made such a categorization, it would be completely different from what it is today. So the third thing I wanted to tell you about before I close, and maybe we can have a conversation, is how ancient DNA has yielded a great surprise with regard to an age-old, more than 200-year-old question, which is, how did the languages that we happen to speak in this country come to be? So in 230 years ago, William Jones, a British colonial justice who was serving in present-day Calcutta, in the city of Calcutta, and had been trained in his school days in Latin and Greek, and was a polylingual, was one of these amazing people who knew many languages. He was interested also in the local scriptural languages. And when he studied Sanskrit, he said the following: the Sanskrit language, whatever may be its antiquity, is of a wonderful structure, more perfect than the Greek, more copious than the Latin, and more exquisitely refined than either, yet bearing to both of them a stronger affinity, both in the roots of verbs and in the forms of grammar than could possibly have been produced by accident, so strong indeed that no philologer could examine them all 3 without believing them to have sprung from some common source which perhaps no longer exists. So this was one of the first articulations of the Indo-European problem. It was not the first, but it was one of the first. And it's the observation that almost all the languages of Europe today, with the exception of Hungarian, Finnish, Estonian, and Basque, and Sami, and as well as Iranian and Northern Indian languages and Armenian are all very similar, are all stem clearly from the same root. Since that observation, there have been ancient texts in ancient Hittite, which was a language spoken in present day in Turkey, in ancient Turkey, about 3 to 4,000 years ago, which show that that too was an Indo-European languages. And there's an ancient language called Tocharian that used to be spoken in the Tarim Basin of China, which was also Indo-European, but is now also extinct. So there's been two leading hypotheses to explain this still unsolved mystery of how these languages got to have their distribution today. Note, these languages have hardly been spoken in the Near East for at least the last 5,000 years. We know that because the Near East is where writing was invented about 5,000 years ago, and very few of these languages, except ancient Hittite and a couple of others, are Indo-European. So one of the leading hypotheses is that Indo-European languages spread with the spread of agriculture, which, as I mentioned to you, was developed 11 to 12,000 years ago and then exploded out of the Near East after 9,000 years ago, almost simultaneously into Europe from Anatolia and into South Asia from Iran. And the idea is that the people who developed agriculture perhaps were speaking these languages, and it was this mass movement of people enabled by this economic transformation that spread language. We know from anthropological studies that spreads of languages in pre-state societies almost always, but not always, but almost always involve movements of people, and this is the kind of economic transformation that allows much more intense use of land, that could bring large numbers of new people into a region. The alternative hypothesis was something known as the steppe hypothesis. The steppe hypothesis of Indo-European origins is motivated by a very different observation, and it's motivated mostly by language. So there are many arguments for the steppe hypothesis, but I'll give you one particularly interesting one. In all of these Indo-European languages, with the exception of ancient Hittite, which is the ancient earliest branching of them, there is a common shared vocabulary for axles, wheels, and some vocabulary for horses. And what this suggests is that the common ancestral population knew of these things, knew of wheels, knew of carts. And we know when those were invented because the archaeological record tells us when wheels were invented. Wheels were invented 5,000 to 6,000 years ago, and we don't even know where they were invented, because once they were invented, they spread extremely rapidly because they were obviously extremely useful. So because this happened in the 5,000 to 6,000 years ago, the suggestion is that the spread must have happened no earlier than that, and therefore it cannot be the spread of agriculture, which happened several thousand years before. So that's the steppe hypothesis. And there's a particular group of people called the Yamnaya. So the Yamnaya are steppe pastoralists who were the first people of the open steppe who used this extraordinary technology of wheels and horses to spread. So prior to the Yamnaya, there were many isolated populations living in the river valleys north of the Black and Caspian Seas. They made different types of pots and stone tools, and each one was different from the other. But after about 5,000 years ago, this Yamnaya group spread and displaced almost all of them. The villages of these groups disappeared. There were almost no more villages. And instead what you see is great tombs only, with very few villages or no villages, often buried with horses or carts inside of them. And the archaeological interpretation is that these people had learned how to take their supplies out into the open steppe, and they were living an ancient version of mobile homes. They were taking their water out into the steppe, and they were exploiting the vast landscapes that had not been exploited before. These were very rich people, their graves were very rich, and they expanded incredibly. They expanded westward all the way to Europe, to Hungary, eastward all the way to Mongolia, running over lots of groups that had come before. So genetically, we were very interested in trying to understand the population movements that occurred during this critical period. And one of the things, the first ancient DNA from Europe was really focused on the transition from hunting, gathering to farming, which occurred between 8,000 or 9,000 and 8,000 years ago. And the first Europeans, all before 5,000 years ago, and the first ancient DNA samples were all mixtures of these two types of ancestry. Blue, which is the hunter-gatherer type of ancestry, and then after about 8,000 years ago, mostly orange, farmers. And there was none of an ancestry that is ubiquitous today, which is green. So if you look in people today, there's a third ancestry type that wasn't there in Europe 5,000 years ago. So we knew in our laboratory in 2014 that this had to have entered Europe sometime. So what we actually did is what in 2012, a couple of years before, a scientist in my laboratory named Nick Patterson had developed a statistical test called a 3-population test. And the way that this test works is extremely simple and it's actually easy to explain, so I'll explain it. So what we did is we took a test population, like, for example, French people today, and we looked at about 600,000 positions in our DNA where we had measured whether people have, say, an adenine or a cytosine, two letters of DNA. And we measured its frequency of cytosine in the French people. And then we compared the French people to all other pairs of about 50 present-day populations we had data from. And we said, on average, are the French intermediate between this pair of populations, the frequencies in this pair, like Sardinians on the one hand and Native Americans on the other. And if the French are intermediate on average, it means mathematically, without any shadow of a doubt, that the French people must be mixtures of at least 2 populations related differently to Sardinians and to Native Americans. So we applied this to each population in our data set, and when we analyzed Northern Europeans, we found an incredible result, which was that French people were intermediate between 2 other populations, and that signal was maximized when one of the populations was Sardinians from Southern Europe, which is actually not a surprise because Sardinians we now view as a relatively isolated remnant population from the first spread of farming into Europe, and so some of the ancestry of French people is from farmers. And the other population of all people was Native Americans. And it was definitely Native Americans. It was not Siberians, not East Asians, not South Asians, not New Guineans, not Australians. And of course, we didn't think that Native Americans had crossed the Atlantic and come into Europe. What we proposed instead was that there was a population that we called the ancient North Eurasians. We made them up with their what we call a ghost population, a population that no longer exists today, but that we can reconstruct statistically based on the patterns that's left behind today in mixed form in present-day people. And that the ancient North Eurasians must have once been somewhere in northern Eurasia and sometime before 15,000 years ago had moved east and eventually contributed to the group that crossed the Bering Land Bridge and moved into the Americas, and people from there had also moved west and contributed to the ancestor of French people. A year later, a group working in Copenhagen sequenced DNA from a 24,000-year-old individual from Lake Baikal in east central Siberia, a boy who was about 5 years old whose dad died near this lake. And that individual perfectly matched these ancient North Eurasians. He was the ancient North Eurasians, much more closely related to it even than Native Americans were. And in fact, indigenous people in Siberia today hardly are related to this individual, which is an example of what I told you before, which is that indigenous people today are rarely directly descended from the first humans who got to that place, but instead are the result of waves of population movement again and again over time. In this case, post-Ice Age repeoplings from the south of East Asian-related people replacing this ancient North Eurasian population in this region. So I'm going to give you a little still movie of what happened in Europe and how this ancestry came into Europe, this green ancestry. Now, first I'm going to show you what this is. So this is the type of data we analyzed. So this is 600,000 positions in the genome that are variable, just like I told you before. Some people have an adenine and some people have a cytosine DNA letter. And this is about 800 individuals from different places in Europe and Western Asia and the Near East. They're color-coded by the populations that they are. And the pattern you see here is that if you actually take this data, so it's 600,000 rows corresponding to the positions we analyzed, and about 800 columns corresponding to the people we analyzed, we multiply this rectangular matrix by itself, and we get an 800 by 800 square matrix, which reflects how closely related each sample is to every other sample. We then perform a mathematical operation on the data called principal component analysis, which shows which separates the samples from each other most efficiently. And the first principal component on the x-axis basically separates all the Near Easterners from all the Europeans. And the second principal component separates northern people from both regions from southern people. So you see these two parallel gradients, and this is done without population labels. We only put the population labels on afterward. So I'm going to show you how this relates to the ancient samples. So I'm going to turn all these dots gray. So here's Europeans, Here's Near Easterners. There are very few populations in between. These are island Mediterranean and Jewish populations, which have known more recent contacts between the Near East and Europe. And if you move forward in time, the first hunter-gatherers of Europe fall beyond Europe in the direction of European differentiation from the Near East. And the correct interpretation of that is that Europeans today are a mixture of these hunter-gatherers and people who are like Near Easterners, but they don't exist anymore today because they've mixed in, as I mentioned to you before. Then the first farmers of Europe are at the bottom of the European gradient, much like present-day Sardinians. They come in from Turkey. Over time, they mix with some hunter-gatherers who are still in Europe, so you see a leftward shift of this cloud. Meanwhile, in far Eastern Europe, you get a mixture of these Eastern European hunter-gatherers and Iranian early farmers who are over here and we now have data from, and you get a mixed population in the far east of Europe, just like you got a mixed population in mainland Europe, but you still don't see people like Europeans today, who are these gray dots, British people and Southern Europeans. They just don't exist 5,000 years ago. But suddenly, 4,500 years ago, they exist. And what happens is people like this, who are these Yamnaya, these people with their carts out on the open steppe, who I told you about before, and these first farmers mixed with each other. And they spread, and they created populations like Europeans today. We now have literally thousands of individuals at this time of transition, and you can see in dynamic detail what happened. So in summary, Europe was massively transformed by two migrations which have contributed the vast majority of ancestry of Europeans today. The first was a mass migration into Europe after 9,000 years ago of farmers coming from the Near East via Anatolia, which is present-day Turkey, and bringing this orange ancestry that formed the vast majority of the ancestry of the first farmers in Europe. And we now have many, many hundreds of individuals from all parts of Europe with this type of ancestry. Over time, there was a resurgence of hunter-gatherer ancestry as the remaining hunter-gatherers living in pockets in the river valleys of Europe or in places that couldn't be farmed slowly over thousands of years interacted with the farmers and were absorbed or absorbed them. And then, after 5,000 years ago, as the Yamnaya spread into Europe, there was this influx of this green type of ancestry, which had never been in mainland Europe before, which contained ancient North Eurasian ancestry admixed within it, and explains what you see today. This type of ancestry now tracks very strongly Indo-European languages, and it's a good bet that it brought some or all of the Indo-European languages spoken today. So I want to sort of conclude the part of my talk where I'm going to tell you about some of these surprises with a very particular case study of Britain published earlier this year, where we published data from 400 samples, 155 of them from Britain, from a culture that's called the bell beaker culture. So the bell beaker culture is so named after these beautiful bell-shaped pots that spread over Western Europe, first in Spain, beginning about 4,700 years ago, and then spread dynamically into the rest of Europe by about 4,200 years ago. But within a couple of hundred years ago, we're in Britain, and Ireland, and Germany, and Czech Republic, and Poland, and southern and northern Italy, all over the place. And it's not just these pots, it's a particular type of buttons and way of burying people, and wrist guards, and a whole tradition which archaeologists often interpret as a religion. These people are contemporaneous to the ancient Egyptians and to the ancient Mesopotamians, and it's a very elaborate ritual. So we actually, initially in 2015, had data just from these individuals from Germany and the Czech Republic, who were from the bell beaker phenomenon. We decided to collect from the entire range. So we now have data from more than 200 individuals from throughout the range of the beaker phenomenon. And our question was the beaker phenomenon practiced by a genetically homogeneous group of people or a genetically variable group of people? And when we made the same type of plot I showed you before, principal component analysis, and plotted the bell beaker individuals, we see they form in two clouds. The Iberian ones from Spain and Portugal are in this bottom cloud, and they don't have any of this ancestry from the Yamnaya. And the other ones from Central Europe have lots of that ancestry. And what's actually happened is that this religion or practice or culture was invented in Iberia, and it spread through communication of ideas or adoption of ideas, much like cell phones spread, where people of very different ancestries use the same practice. But once it got into Central Europe, it spread further through migration. And I'll show you evidence of that. So in Britain, where we have data, we reported data on 155 individuals from 6,000 to 3,000 years ago, you can measure the proportion of ancestry from first farmers of Britain who arrived in Britain about 6,000 years ago. It took them a couple of thousand years ago to get from Turkey to Britain, to a couple of thousand years, and steppe-like ancestry. And when you look at the data, you see that until 4,500 years ago, all the ancestry of everybody, without exception, is first farmer. And then bang, at 4,500 years ago, everybody has about 90% ancestry from the continent. And it's a minimum 90% population replacement. Shortly before 4,500 years ago, the last stones of Stonehenge went up, and these first farmers had built it, and within 100 years, they didn't know that they would be replaced by these people from the continent who very shortly ended the practices that were common at places like Stonehenge. So the implications for Indo-European origins is that it falsifies the single best argument in favor of the Anatolian hypothesis, the idea that it spread with farming. And the best argument was that farming is the only economic transformation that could plausibly enable the large-scale movement of people into Europe. There couldn't have been anything later that was equivalent that could have done this, because once a dense farming population was established in Europe, it would be very hard for anybody else to make a dent. Kind of like the British didn't make a dent in India, even though they controlled it for a long time, demographically, because there was just intensively practiced agriculture there already. And yet we know something happened, right? So I don't know how it happened. I don't know what mechanisms allowed these new people to achieve this large-scale replacement, but it did. It clearly did. And it's a 70% replacement in Germany, a 90% replacement in Britain. And now it's up to archaeologists in dialogue with geneticists and anthropologists to figure out what's going on. So to conclude, what's happening right now is this is a map of where the ancient DNA data are available from. It's very Eurocentric, not because Europe is a more important place in the world than other places, but that's just because that's where the studies were first done. And there's also the samples don't really go until stop mostly before 3,000 years ago. Really, the DNA that we have is mostly between 10,000 or in some cases 100,000 years ago and 3,000 years ago, and it stops after that point. So the question of how people like we are today got to be the way we are today over this incredibly important period in the last 3,000 or 4,000 years is not answered by ancient DNA. So this field is exploding dramatically. We're learning many, many new things all the time. Every time we get DNA from an ancient archaeological culture we had not sampled before, we find things that are extremely surprising to the people who had studied that before and to us. And so it's a very exciting time to be working in this area and to be dialoguing with archaeologists and anthropologists. And it's all happening right now. So I wanted to say that ancient DNA is teaching us humility. Much of what we thought and what I thought myself coming into this area was wrong. And it's an unusual field where scratching the surface is guaranteed to surprise. I didn't want to write this book for a long time because people kept saying someone should write a book about this, and I can't understand all the papers, they're all very technical. And I kept saying no, and the reason I said no was because I just wanted to focus on the research. But increasingly, my colleagues came to become archaeologists and anthropologists and linguists who really just couldn't grapple with this work because it was all written in jargon. And so I thought I could write a book that described what's going on and write it in a language that was not using our jargon, but that could communicate to those people, and at the same time try to write it and that meant I had to write it accessibly to sort of interested people who would want to learn about it. And so that's the book that emerged. I ended up writing this book, and hopefully it will be useful to anybody who's interested. Thank you. Question back there.
Yeah, in your very first slide, you had Neanderthals on the one side and Cro-Magnons on the other. But then you never mentioned Cro-Magnons or talked about them again. Is there a reason?
Right, so I was contrasting these two big-brained humans who lived in Western Asia, Western Eurasia, and in Africa around the same time. Cro-Magnons are examples of anatomically modern humans, the same people who spread into East Asia, into South Asia, all around the same time. But they're some of the earliest skeletons. A lot of this field is very European-centered just because the first well-described skeletons from them, but they're just examples of of anatomically modern humans, the first ones out of Africa. Question back at the very back.
On that subject, if somebody's not 100% Neanderthal or 100% Denisovan, then they are mostly out of Africa, right?
Correct.
Okay.
Correct. So the truth is that I don't want you to come away from this talk thinking that Africa is not our primary homeland. It's overwhelmingly the primary homeland of everybody living today, not just within the last 50 to 100,000 years, and actually more anciently, again, 2 million years ago and so on. But I did this sort of extreme pendulum swing toward thinking everything is out of Africa. There's no contribution to non-Africans today outside of Africa was too extreme. It was sort of just a comment. It was almost an orthodoxy 10 years ago in genetics. There's a question over here.
Because of the Ice Age and where there was habitable warm land, did the Neanderthals originally start from Africa too?
So humans first migrate out of Africa for the first time a little bit after 2 million years ago. So Homo erectus, a type of human, appears approximately simultaneously from earlier Homo forms, which are all in Africa, about 1.8 million years ago, both in Africa and outside of Africa. And ever since that time, there have been lots of humans in Eurasia. It's a rich habitat full of humans. The question of where the common ancestors of Neanderthals, Denisovans, and modern humans lived, and we actually can estimate from the genetic data when they lived, which is probably about 500 to 700,000 years ago, is probably Africa, but it's actually not surely Africa. So the genetic data has made it plausible that the lineage at the time that those three groups were born actually was somewhere in Eurasia, and there was a back migration of this group to Africa where the ancestors of anatomically modern humans incubated, then spread again out of Africa 50 to 100,000 years ago. So there's this pendulum swing toward thinking everything is Africa, the Australopithecines and Lucy and the African apes, and it's clearly true before 2 million years ago, but between 2 million and about 500,000 years ago, there's what the anthropologists call a muddle in the middle. There's equally rich skeletal records, but not very rich, both simultaneously in Eurasia and in Africa. And it's not clear, and that there's sort of parallel movement toward larger brains. Maybe there's multiple movements between Africa and outside of Africa. Maybe there's parallel evolution. But in fact, this idea that it's just out of Africa again and again and again and again and again, there's no reason it has to be that way. It probably is that way. Over here.
So, sort of, I guess, two questions, a little bit related with the Indo-European movements. So, is there data similar to the genetic movements into Eastern and Central and Western Europe, down to India and Iran and those places from the steppe? And then I guess as a second question, is there any genetic evidence, sort of the region, the Pentoharian, of this steppe ancestry and people who now live in the Tara Basin, or is there too much recent mixture to say?
So that's a great question. So, in fact, we now know in the last 4 months an incredible amount about South Asia and about how steppe ancestry related to the Yamnaya was pumped into South Asia and when exactly that happened. So South Asians today have about 0 to 30% ancestry ultimately from the Yamnaya, these steppe pastoralists. It's higher in proportion in people who speak Indo-European languages, which are the languages of the north of India, than in Dravidians. And in fact, it's relatively higher proportion in Brahmins from northern India, who are the ritual practitioners of and custodians of the Indic Sanskrit texts, than it is in other groups of overall similar proportions of West Eurasian-related ancestry. What's very clear now, because we have genetic data from Central Asia, from Uzbekistan and Tajikistan and Turkmenistan, is that steppe ancestry from the Yamnaya doesn't even appear at the gateway of India until about 4,000 years ago, when it suddenly appears around these towns of this ancient civilization called the Bactria-Margian archaeological complex, which was contemporaneous with the Indus Valley civilization to the south in what's present-day Pakistan and Western India. And then in these cemeteries, we have hundreds now, or sorry, not hundreds, but many, many dozens of individuals from these cemeteries who are genetically broadly quite like early Iranian farmers. But in the cemeteries, there are outlier individuals who are not, and these people at first are like the hunter-gatherers of Siberia and that region, and then later they have steppe pastoralist ancestry. So what's clearly happening, and there's archaeological evidence for this as well, is there's interaction between these steppe groups that's now entering this region, and then within 500 years we see it pumped into South Asia, and contributing about 30% in some cases of the ancestry of South Asians today. And so we have a very precise understanding of when this ancestry came in. It came in about 1,000 years later, or 700 years later than it did in Europe itself, via successor cultures of the Yamnaya that were spread over the eastern steppe. So regarding the Tocharian question, which is the Tarim Basin, where these languages are attested thousands of years after the events I'm talking about in ancient texts that are preserved due to the dry conditions, there's several pieces of evidence relevant to this. One is that individuals from the Altai Mountains, from an ancient culture called the Afanasyevo, which is contemporary to the Yamnaya, which have many different burial traditions, in fact, are genetically identical to the Yamnaya. So what's very clear is that the Yamnaya spread that far east basically to the threshold of this region, and they changed their culture, but they were there. Another thing is that there's sporadic evidence in populations from later in the period of Yamnaya ancestry without admixture from the type of ancestry that appears later in the steppe and actually gets into India itself, that you see in this part of East Asia and Mongolia, and is consistent with the idea that an early branch of this population, Yamnaya, before some of the events that later impact Iran and India get to this region. So until there's data directly on a chain of transmission in this region, we can't know for sure, but it seems likely that Tocharian, which is the second oldest branch of Indo-European languages after Hittite and other Anatolian languages, is an early offshoot of the Yamnaya spread, and that Hittite itself is prior to the Yamnaya spread. This question back there.
Yeah, do you have a feeling as to how many other ghost populations of other human, and also the ability to, in your ancient DNA, how good can the technology become to, how far back can you go?
The question is, how many other ghost populations are there?
Might there be?
I think there are hundreds. We already know many others. So in fact, there are many other cases where, for example, we have predictions of populations not yet found in the ancient DNA. What's clear from looking at genetic data, not just in Europe, but other places that we and others are beginning to get data from, is that people are the results of layers and layers and layers of mixture. That nobody's pure, and that in fact, those ancestors were not pure either. That in fact, people are just the results of mixtures and mixtures and mixtures. And the right way to look at this may not be that of a tree, but as a trellis, a kind of just layering and layering of mixing. And so the other question was about the technology and if it's getting better. The technology is continuing to get better. Just a couple of years ago, there was a discovery, for example, that a particular part of the body, the skeleton, contains up to 100 times more DNA than other parts of the skeleton. And much of the explosion that you're seeing is due to this realization that this inner ear bone, the oda capsule that contains the cochlea of the inner ear, is somehow a DNA-rich mother lode. But there's also other just technical innovations related to efficiency of extraction of sequences from the bones that we obtain that themselves have been just as important, and that combination of things has made it much more efficient. It seems likely there's a few more tricks up the sleeves of the technicians who are developing these, and I think the technology will continue to get better, but really a lot of the work now has to be in applying these incredibly powerful technologies, which are really ready to be applied all over the world. Question here.
This seems to be a huge blow to racism. So, that people are really mixtures of everything, a lot of other things. And also, I was thinking about why people go to war and why people fight with each other. And it seems like people took over other people for like centuries and millenniums, and that this may explain why some people have such conflict. I mean, sociologically, over many, many years.
I think the observation was that this really destabilizes our understanding of the structure of the world, and if you have an idea, pictures that people have in their mind and that they're taught as children or come to understand as children of what, quote, races are, I think that this destabilizes it. For me, it very much does. I think it's hard to think about the population structure of the world, for me, the way I learned or intuitively learned as a child. I also think that this gives us a sense of what happened in the past and the nature of what happened in the past and the drama of what happened in the past. I mean, if you look at Britain 4,500 years ago, that was a very dramatic event. So these people built Stonehenge, and then within 100 years, their descendants were largely not contributing to the population. It can't have been an easy experience. We actually are about to publish some data from Iberia where the replacement was not so dramatic. It was about a 40% population replacement, quite big, but in Iberian Spain. But it's 100% male replacement. And so what you're actually seeing is that the DNA of the population gets 40% replaced, but what's clearly happened is that the males coming in had preferential access to local females, and new males coming in had preferential access to the female offspring of those unions. And so what you're actually seeing is must be a very complex, difficult interaction between the incomers, and you can see it written in the DNA. You can see something about the nature of these past events. And so you're saying that this teaches us something about conflict and the difficulty and the inequalities that have existed in the past. And it's really amazing that we can learn about this in DNA. There's so many examples of that nature. I could go on and on and on and on and on. So,
No wonder people fight.
yeah, this question here.
Yeah, a clarification, and you may have said it, and I was just trying to catch up. When you talked about taking the French population, could you clarify if you mean like living today or a skeletal French population, and how did you define that? Like how many years would they have had to live there? So many people who live in France there didn't live there 15 years ago. How do you define that, and how do you deal with that issue of our modern transitory nature?
So the question is, what do you mean by French, right? So, and that's an excellent question. And French means different things to different people at different times. So what we technically meant here was the people who were identified as French in a data set of data that we were given from present-day people. So this is a data set that was generated in the early 2000s, a set of DNA samples from 51 diverse populations around the world of present-day people before ancient DNA technology was available. So I'm talking about modern French. And I don't know which particular region of France this is, and I don't know whether they were sampled in villages or whether they were sampled in Paris. I don't know whether they were sampled in Marseille or Bordeaux. There are differences in each place. It happens to be the case that when we run the same test on British or on British or on Germans or on Northern French or Western French or Eastern French or Southern French, which we now have data from all of these places, we see the same pattern. And the reason is that this is a broad Northern Central European pattern that is shared powerfully amongst these groups. But the question of how to define a population and what a population means and what are sharp boundaries and what are graded boundaries is an incredibly important one. So technically, when we analyze data, we draw a circle around a group of people in a variety of ways. We try to look genetically at the data and see if they're relatively homogeneous relative to other samples in our data sets. We might pop out some outliers who don't look homogeneous, but there's a good sort of epistemological or philosophical question about what we mean by a population, and it's a little bit hard to define. But in practice, it's the group of people we happen to have analyzed. So this group of people has this ancestry, and what we're saying is that the group of people from France who ended up in this data set, we know that they are a mixture of two groups, one of which is, at least two groups, one of which is related to Sardinians and one of which is related anciently to Native Americans. We can say that about these people. Now, how representative these people are about France more broadly will require dense sampling of people from present-day France and perhaps the same region. But I think that we have an increasing sense of the degree to which ancestry changes at regional scales in different parts of the world, local regional scales as well as over time. Question over there?
In your data, can you infer anything about function, like the Stonehenge thing is so dramatic in a short period of time? Does that line up with any kind of disease resistance factor or fitness factors that could explain the genetic difference?
The question is why these people were able to make such a major impact in Europe coming from the East. So there's an incredibly interesting observation that is very hand-wavy at this point. And the interesting observation is the following. So in some of these steppe samples from 5,000 years ago, taken from teeth, the DNA from teeth was taken out and the DNA was extracted. And in addition to the human DNA, there is also other DNA. Soil DNA from the burials, but also pathogen DNA from microbes that infected those individuals when they died. And about 7% of the individuals in this initial survey had DNA from Yersinia pestis, which is the Black Death pathogen. So if people have Yersinia pestis in their teeth, it probably means they die of it because you're not normally bacteremic. And so what that's telling you is that a minimum of about 7% of the 100 people in that sample from the steppe about 5,000 years ago, or 4,000 or 5,000 years ago, were infected with and dying frequently of the Black Death. Now, when you look at the sequences obtained of this Black Death, it's not bubonic plague of the type that you see in 1347. It's missing the chunks of DNA that are necessarily for flea and rat transmission, but it contains the transmission factors that are necessary for pneumonic plague, which is a inhalation-transmitted plague. And it's quite clear that this was killing a lot of people 4,000 to 5,000 years ago. And one idea is that this was an endemic plague on the steppe, and European farmers were naive to it, kind of like Native Americans were naive to European diseases, and that perhaps these reached Europe. I'm now waving my hands, before the steppe people got there, and there was like this or other plagues caused a lot of dying off of the population, and it was more difficult to stand up and resist the movements of these people. So that's the type of thing that you might begin to be able to learn from the genetic data. The archaeology shows that there was a decline of the population in the centuries before the arrival of the Yamnaya and their successor groups that carried this type of ancestry known as the corded-ware complex. And so I think that one of the things archaeologists are now intensely doing is trying to understand which factors are important or not in this process. There's a question here.
So I may be among some of us here who came to your work from your New York Times op-ed, and I read it as a kind of cri de coeur to sort of convince the rest of us that there was some very important lessons to be learned from your work. I wonder if you make the connection between the work that you did to produce this book and your reasons for writing that New York Times op-ed.
Right, so the op-ed article was really a piece of chapter 11 of the book, which is called The Genomics of Race and Identity. And it's not the main focus of the book, and so I was actually conflicted about whether to write this op-ed at all because I thought that it might create a lot of interest and distract from what I actually wanted to be the main focus of the book, which was this incredible revolution in our understanding of the human past. But I decided, for a variety of reasons, to write it because I thought there was something to say. And what I was writing about in this op-ed was that our emerging understanding, which is not mine, and it's not solely based on ancient DNA, it's really based on the medical genetics and genomic revolution about the nature of human population structure, makes it clear that the population we live in today is actually quite diverse. The human populations today are the result of groups that have largely diverged from each other for, in some cases, 200,000 or 300,000 years, which is easily enough time for major changes to occur in populations under the influence of random factors or natural selection. And so and modern medical technology is incredibly powerful and are measuring genetic differences across individuals that are clearly also going to be slightly different, although not very different across human populations. And the way that anthropologists and geneticists have talked about this work have really summarized the genetic structure of human populations to say we're all incredibly similar to each other and there's no space for substantial differences amongst human populations. There's no room for there's no meaningful biological differences on average across human populations. It's simply not true. There is room for it. They're small. They're on average 6 times smaller than the differences amongst individuals. You see how different people look in this room? Well, on average, populations will probably differ by 6 times less than that on average. But that's not nothing. And we have to come up with a way to actually talk about those differences rather than pretending they don't exist. It used to be the case that we could pretend that they didn't exist because we never measured them, and we couldn't measure them, but now they're being measured, and even if it was outlawed, this type of research in the United States or in Europe, it's going to go on in China, and once these discoveries happen, there's going to be no turning back. And so I think we need to have a more sophisticated way of talking about that. And if we geneticists don't talk about these issues, which we haven't been doing because we're so anxious about talking about these issues, that vacuum that we leave is going to be filled by pseudoscience, by pundits, by people making stuff up, because we're not fulfilling our role as scholars of providing guidance about the topics about which we're expert to the public. It's not an easy issue, but we need to learn a way to talk about this because it's upon us now, and so that's what my article was about. I honestly have no idea what the nature of the differences will be amongst human populations. I'm quite confident they'll be very modest, but I have no nature what they're going to be. I think it's highly unlikely that they're going to conform except in random cases to conventional stereotypes, but we're going to have to learn how to deal with them, and so that's, I think, what my article was about.
I thought I was pure French from generation to generation. Now I always say I am pure. So are you really disappointing me? And I say my children are a mixture now because I married an American man, which is Holland, English, bunch of. So I say so am I not pure French?
It's whatever you think.
I should have stayed home tonight. I'm not pure.
Thank you for talking about this, especially in a way that us lay people can understand. So my question is, how do we keep up after your book with the new information? Do you have any recommendations on how we can follow what's going on in research like yours?
How do you follow what's going on? So this work is, it's a good question. I think the book attempts to be a good introduction for anybody who's interested. It's a serious book, so I tried to write it to be comprehensible. But it's not. So it should be understandable to anybody who's interested in engaging. I think the book gives a flavor of all of these topics, but if you want to know exactly what the findings in this field are, you probably have to pick a few journalists who you trust, like maybe Carl Zimmer or something like this at the New York Times, who's a better journalist and who's more serious, and try to read their articles on this topic. And maybe what you have to do is look at some of the blogs. There's a few sophisticated blogs that try to follow this work and translate this work. And maybe other people will write similar such books. I'm pretty exhausted right now. The question over here.
Just to follow up immediately on that, what blogs would you recommend that are out there?
So the blogs are complicated. I was a little bit reluctant to suggest them because they basically, there are some quite sophisticated bloggers who are consuming and translating and actually analyzing this genetic data. And they're very political, some of them. And in general, they lean right. And they often lean right. My interpretation is that a lot of these people have looked at the genetic data because we make all of our data publicly available. They've looked at it and they basically think, they respond to statements that you hear from anthropologists that there are no meaningful biological differences and that we're all the same. They look at the data and they say, no, it doesn't look like this at all. There's profound differences amongst populations, and they kind of feel the academics are pulling the wool over our eyes. And a lot of these blogs have this flavor of people are pulling the wool over our eyes, and here's the truth, I'm analyzing the data. However, there's very sophisticated people, Razib Khan and someone named David Weslowski and others, who are like seriously looking at this data and seriously in touch with the data and writing about it in this fairly accessible way. But if you look at the blogs, you might, depending on who you are, you might feel more comfortable or less comfortable with the way they talk about it.
So was the latter person you mentioned, the author of the Eurogene?
Yeah.
Blog? Yeah.
Question here.
I just have one question. So obviously, looking at, I assume most of these differences are in non-coding regions, there's just SNPs. Would you find differences if you actually analyze functional genes, like master transcription factors, something like that? Would something different come out of these analyses?
The question is where our information is coming from about human history. Most of it's in what we call junk DNA, random bits of DNA that are floating between the genes, because that's the vast majority of our DNA. And would it be different if you look at important sections of the genome that actually do stuff like make genes or affect the function of genes? And the answer is, for these patterns are broadly supported by all parts of the genome, including when we subset on those parts of the genome. But there is sometimes profound natural selection over time, especially on functionally important parts of the genome. And I think one of the things that's going to emerge over the coming years, or year, is much more discoveries, much more information about the nature of the biological change that's occurred over time. What ancient DNA does is it's a time machine. It gives us information about before and after events like urbanization or the invention of agriculture, what happened biologically to a population before and after these profound lifestyle changes, and we can watch the population react over thousands of years to this. This is a very powerful instrument. When you are in a laboratory, you like to do time series experiments where you take a mouse and you do something to it and you watch the mouse as it reacts to that thing you've done to it. And we can't do that with people. But actually now we can, because we can actually look before and after key profound lifestyle changes and at least get some of the types of information that you would like to get from a time series experiment. So I think that that's something incredibly powerful. The promise of ancient DNA was in two areas when it started. One was on understanding human migrations, and the other was understanding how we've changed biologically over time. And it's been a runaway success in terms of explaining population migrations and mixtures, and continues to be, and it's only at the very beginning. But we hardly know anything at this point about biological change over time. That's not because it's not actually promising, but because the way to actually learn about that is by obtaining large sample sizes, large enough to watch frequency changes over time, and that's not been available until basically the last year. And now we have those sample sizes, and we can begin to actually learn how the frequencies of different things that we know are biologically important based on powerful studies in present-day people, how they've reacted and changed over time in response to evolution.